Hi there, this is David Nguyen, Principal Investigator at the Tissue Spatial Geometrics Lab. What I do is I write algorithms to measure spatial information in the structures of tissues and tumors, and in this presentation, the structure of sugar molecules known as glycans. The information in how cells are arranged in tissues and tumors and how sugar molecules are arranged in glycans are multidimensional codes, unlike the information in DNA. Therefore, we need new tools to measure this information in order to quote-unquote crack them. Glycosylation is the addition of sugar molecules to proteins and lipids, and glycosylation is involved in almost every aspect of how the cell works, from protein stability to cell adhesion, all the way to signal transduction. We know that the structure of the glycan determines what happens to the protein and how the protein behaves. So we know that there is a glycan code, but we have yet to quote-unquote crack that code. One of the reasons that it's been difficult to crack this code is because it is multidimensional as opposed to DNA. The code in DNA is linear, meaning it's a one-dimensional array of symbols, and it's semantic, meaning information is codified as a linear sequence of characters. The glycan code, however, is nonlinear in that it consists of 3D branching array of symbols, and it's non-semantic, meaning information is codified in multiple dimensions. Therefore, in order to translate, like as in a language, a 3D code, we need a multidimensional approach. I'd like to do a quick review about the first method, which is called the distance and connectivity matrix. I will not go into the details of that one here because there is a video you can watch on our blog in the link at the bottom of the page. But I do want to say that that first method was designed to capture information about the distance of each node to the base node and the degree of multi-connected nodes within that glycan. However, there are a number of key features of glycans that it could not capture, such as the number of monosaccharide types, the types of linkages that connected those monosaccharides, the number of linkages, and whether different monosaccharides were connected to each other. Thus, I created a second method, which I will talk about today, called the position and composition matrix. This one measures these features and has an added advantage that the matrix was designed to be vectorizable. That is, you can stack them into one column of data, such that if you combine multiple columns of data, you can apply data science methods in a way that is meaningful. Notice here that for the DC index, the answer is not yet in terms of the vectorizability of the matrix, because I do plan to update this into version 2, which will allow us to do that. In a matter of a few minutes, you'll see why this is a very advantageous feature for the method. Here's a summary of the position and composition matrix, which I will not read. You can pause the video to read if you want. Here are the rules for applying the PC matrix to a glycan, which I will not read because I will explain it to you through an example. So here is an example of an N-Glycan. And for the PC matrix, what we do is we divide it into layers, where each layer is separated by only one glycosidic bond. Therefore, this fucose added to the base node actually belongs to the second layer. After we've divided a glycan into layers, we then measure these 5 features for every single layer. First is the number of monosaccharide types in that layer. Second is the total number of units regardless of type. 3 is the number of interlayer linkages. That is these lines crossing the dotted line. And then 4 is the number of linkage types, because there are unique types of bonds between these monosaccharides. And number 5 is the number of monosaccharides connected to a different species in the previous layer. As you can see in layer 3, this mannose is connected to a glucnac in the previous layer, while this mannose in layer 4 is connected to an identical mannose in layer 3. By measuring these same 5 features for every single layer, what we get is a data matrix. For example, I will walk you through how we derive the numbers for layer number 4, which is right here. So the first feature is the number of monosaccharide types. For layer number 4, there's only one monosaccharide type, that is, they're both mannoses, and so the first value is 1. The second feature is total number of units. Layer 4 only has 2 units, so the number is 2. Next feature is the number of interlayer linkages. Well, layer 4 has 1, 2, 3, 4, and 5 total linkages, extending from itself to the layer above and below. So the answer is 5 for the NAL feature of layer 4. Next is the NLT, which stands for number of linkage types in layer 4. Well, layer 4 has an alpha 1, 2, that's one type. It has two alpha 1, 3s, which is a second type. And then it has two alpha 1, 6s, which is a third type. So therefore, there are 3 linkage types in layer 4, and so the number here is 3. Lastly, feature number 5 asks, are any monomers in layer 4 connected to a different monomer in the previous layer? The answer is no, because layer 4 has 2 mannoses. They're both connected to also a mannose in layer 3. So the last value in layer 4 is 0. So in this way, we systematically measure the same features for every single layer, and we have to make sure to measure the features in this exact order. Otherwise, we won't be able to compare these matrices objectively with other matrices later on. So if you want to derive a scalar index from this data matrix, what you can do is sum every single column and then sum all the results of those columns to get what we call the PC index, which is one value, right? A scalar value that is a simple number to represent the information in this glycan. This diagram is just to remind people that each layer is separated by one glycosidic bond. So though this fucose attached to subunit number 9 is typically drawn on the same level, because it is separated by one glycosidic linkage, this fucose actually belongs to layer 6, right? And this fucose up here, number 19, belongs in layer 8. If this fucose happens to have another monomer attached to it, then that one would belong to layer 9. The reason I made sure to measure these features in the exact same order for every single layer is because we can then stack every single layer underneath the one that came before it, which is called vectorizing the matrix, turning into one row, or in this case, one column. Once you do this, you can then combine the vectors of multiple glycans into a data table. And the data table will have the exact same order of features in every single layer. Thus, when you do dimension reduction techniques on this data frame, the results are actually meaningful as opposed to meaningless and not reproducible. My hypothesis is that the systematic approach of the PC matrix will result in structural themes clustering together after vectorization and dimension reduction. So to test this hypothesis, we first analyzed the core vertebrate N-Glycan synthesis pathway. So vertebrates have a core glycan that looks like this. It begins with the first monomer, and then 13 steps later ends up in its final form. We analyze each of these 14 structures using the PC matrix, and then first we plotted the PC index for each step. And as you can see, the index forms a relatively straight line with a rising slope. Now, while this rising slope does correlate with what we see, that is the complexity of the glycan increasing, it's a very crude representation of the increasing complexity because we know that there are multiple ways in which the complexity is increasing. First, you have the branching patterns, and then you have these third type of monomers being added at the end. But all of these changes are only represented by a straight line. While the PC index is convenient as a scalar value because humans just like single individual numbers, a lot of information is lost when we do this, which is exactly why I made sure to define the PC matrix as having a very specific order in which to measure the features, an order which must be maintained for every single layer, because when we vectorize each matrix, we can combine them together in a meaningful way to do dimension reduction. And that's exactly what we did. We did principal component analysis on the vectorized matrices of these 14 structures. And what we found is that the information within them is anything but linear. Though this pattern looks like an S shape, when you view it in 3 dimensions, it actually forms a spiral. As you can see, this multidimensional way of measuring the information within these structures is much more nuanced. As you can see, different structural themes clustering in different parts of this three-dimensional space. And as a scientist, it is very satisfying to see the data being so elegant, because we know that nature is very elegant in how it operates. And when we measure nature in ways that are more elegant than just a straight line, we know we are on the right path. So the PC matrix is novel because it translates the glycan structure in a way that incorporates spatial information. It is objective because it has systematic rules that anyone can follow to get the same results. And it is quantitative, so that allows us to do statistics to extract more insights. So next we wanted to apply the PC matrix to more complicated glycans, and we decided to analyze this paper by Suzuki et al. They extracted N-Glycans from chicken colon, and we picked this paper because they provided a legend that allowed us to reconstruct every single glycan that they reported and know exactly what the monomers were and what the linkages were. Here are examples of how we divided up some of their glycans into layers. 3 people independently did these calculations before comparing the answers for accuracy, because the calculations we do for these molecules will be part of how we test the accuracy of the software that we're developing, which will automatically do the calculations for you. We applied the PC matrix to 25 glycans from this paper. We vectorized their matrices and then did principal component analysis. And this graph shows how they scatter. From this, we know that the PC matrix is unable to tell the difference between isomers. For example, here, these D structures are only different in the location of the sialic acid at the end of these branches. And to our satisfaction, we found that the structures grouped just as we had hoped. In this red cloud at the top, we see structures that have a fucose added to the base node. And then going counterclockwise, on the left, in the blue cloud, we see structures that have a bifurcation and one arm longer than the other. Next, we have a region down here that represents glycans that have multiple bifurcations. For example, here's one bifurcation and here's another one. Then in the purple region, we have glycans that have bifurcations and trifurcations. For example, here's a trifurcation, 3 branches, and here's a bifurcation, 2 branches. Lastly, there's a green region in which the N-glycans look like O-glycans. And this was very exciting because this tells us that the PC matrix will likely work on O-glycans, which is the next class of glycans that we want to numerically translate. We have shown that even in the case of complex glycans, the PC matrix can separate them in meaningful ways. Another feature I like to point out about the PC matrix is that there is a subindex we can generate that might be useful for studying the effect of fucosylation at the base node. So N-glycans often have a fucose monomer added to the base node. These 2 structures are identical except for that fucose. When you calculate the PC matrix, the addition of the fucose only affects the first 2 layers. So if we add up the layer scores in the first 2 layers, what we get is what we call the base node fucosylation index, or BNFI. So structure A has a BNFI of 9, while structure B has a BNFI of 16. Well, 16 minus 9 is 7 units. What is the significance of these 7 units? Well, 7 units is 14%. That is 7 divided by 49. 14% of the information within structure A, which is without the fucose, meaning one fucose can add a sizable amount of information to the glycan. And this is important because the effect of one fucose at the base of this structure has an effect that is not linearly proportional to the mass of the entire structure. Thus, we need a metric to measure its effect in a way that's not 1-to-1. We hypothesize that this index may be useful in determining the effects of a base node fucosylation in the future, and we look forward to testing that hypothesis. In conclusion, the PC matrix captures spatial information within N-glycans in unique ways, and it has the potential to elucidate the quote-unquote information states in glycobiology, as opposed to just counting the abundance of physical molecules, to reveal the grammar of the glycan code. I will now talk about work in progress. As mentioned earlier, we are developing a software that allows people to draw any glycan and specify the type of linkages. And this software will automatically calculate the matrices and the indices of interest. And the person leading this is Daniel Gallegos, and he will be applying to MD, PhD programs in the next 12 months. So you or your colleagues may be interviewing him. Our other collaborators are Aurora Britannia Diaz Fernandez and Bruno Pavletich, who want to apply my methods to studying glycans in infectious disease. I won't go into the details because they will speak about it themselves next.
Okay, I will also be brief on the some applications, potential applications of the PC and DC indices that they've proposed. I'm Bruno Pavletic. I'm a PhD student at the Technical University of Braunschweig, and Aurora, MD and a master student at the Universidad Europea de Madrid. Okay, so Dave, you can go next slide. So first project that we kind of propose to make use of the glycan DC and PC indices is validating them, taking downloading the database of glycans, which is found on online, and seeing is there any biological significance, and evaluating the biological significance by grouping the glycans and calculating their DC and PC indices according to their biological functions and biological groups where they can be evaluated. So first downloading the database, and the idea is having making a tool for to use make use of the those indices as a low computation, not a computation intensive way to try to predict or associate the functions of novel glycans. If a person finds a new glycan, discovers a new glycan, then can they associate the biological function based on DC or PC index? Of course, go in the right direction. You can go to the next slide, Dave. Thank you very much. So the idea is so for, as I said, like downloading all the glycan structures, 2D structures from ArcSDB database, and then plugging them into the program to calculate the PC and DC indices for every glycan from the database, and try to see, first of all, are there any significant differences between groups. Here you can see like the idea of the data. The work has not been done, but you can see the schematic representation and the idea of hypothetical results. So what we could expect, or maybe not. So this is still the question. Determining the statistical significance between the different groups, and doing the PCA analysis between those different groups to see, do they cluster, do the separate groups cluster separately, and also in addition to that, comparing the differences between DC and PC indices among different glycans to their 3D structure. So how do 2D comparisons, how do they compare to the 3D differences in glycans? Okay, next slide, please. And this once this is done, we propose a project of actually applying then something like that. So we wanted to research whether there is any difference in PC and DC index in different strains of SARS-CoV-2 virus, because this it's well known that the coronaviruses have heavily glycosylated spike protein, the spike receptor, and what is more, have a different level of their receptor binding receptor protein, and there we wanted to correlate, are there or see just explore if there are any correlations with the distributions of with glycan diversity distribution across the protein, and could we calculate based on the DC or PC matrices, can we group the strains together and see if there is any correlation or anything significant considering when we consider the PC and DC indices. Okay, next slide. So here we just show from one paper. Here is the glycosylation profile of various different strains, and the idea is taking of the spike protein, and the idea is taking all those different glycans and see are there any differences in average DC or PC index, and determine does it correlate with the virulence. Next slide. And with our final slide, so we can calculate the distribution of the indices across different SARS-CoV-2 variants on the spike protein, and correlate it with infectivity or immunogenicity. And we are still brainstorming all the possible future applications, but this is kind of, let's say, first project to test out the whole idea. Thank you very much for listening.